Our third line of defense is the body's ability to defend itself against specific invading things, pathogens such as bacteria and viruses, toxins, and even foreign tissue. So this is called adaptive or specific immunity. Substances that are recognized as foreign and provoke an immune response are called antigens. That simply means antibody generators. So antigens are foreign substances that cause the immune system to respond by generating or creating antibodies. Two properties distinguish specific or adaptive immunity from innate immunity. One is specificity for a particular foreign molecule or antigen. So this also involves distinguishing self from non-self molecules so that the immune system can respond to pathogens and not to self cells. And the second characteristic that is different is memory. So for most previously encountered antigens, there is a memory of what the immune system did to destroy that pathogen. And then on the second encounter with the same antigens on the same pathogen, we have an even more rapid and effective immune response. And a lot of times that gives us what we call immunity. So we don't get sick or as sick in that second encounter. Another characteristic of the adaptive immunity is that it is systemic and not localized, as we saw with some of the first line of defense. The two primary types of adaptive defense are humoral immunity, which is mediated through the B cells, and and cellular immunity, which is mediated through T cells. So we'll look at each one of these types of cells and their role in this type of immunity. So let's begin by discussing a little more about what adaptive immunity is. So adaptive immunity involves two of the types of lymphocytes, the B cells and the T cells. Remember when we discussed leukocytes, lymphocytes were T cells and B cells. So as we continue to get into this adaptive immunity, we'll look at the role of each of these. But both of these cell types develop in primary lymphatic organs. Remember the red bone marrow and the thymus, and they develop from those pluripotent stem cells that originated in the bone marrow. B cells complete their development and their maturation in the red marrow, and that process continues throughout life. T cells develop in the bone marrow, but then those immature T cells migrate from the bone marrow to the thymus, where they mature. Once an immune response has been activated, each type of lymphocyte forms a clone of cells that can recognize a specific antigen. And when these cells are creating clones, they are proliferating or creating more and more and more of those specific cell types. So remember, adaptive immunity has two types, cell-mediated immunity and antibody-mediated immunity. Both types are triggered by antigens. In cell-mediated immunity, cytotoxic T cells directly attack invading antigens. In antibody-mediated immunity, B cells are going to differentiate into plasma cells, and then those plasma cells are going to make and secrete specific proteins called antibodies. An antibody can bind to and inactivate a specific antigen. Cell-mediated immunity is particularly effective against intracellular pathogens, and that can include viruses, bacteria, a fungus, or things that are inside cells. It's also very effective against some cancer cells and foreign tissue transplants. Cell-mediated immunity always involves cells attacking cells. Antibody-mediated immunity works mostly against extracellular pathogens, and that can include viruses, bacteria, or fungi that are in body fluids outside cells. Since antibody-mediated immunity involves antibodies binding to antigens in body humors or fluids, such as the blood and the lymph, it's also sometimes referred to as humoral immunity. When a specific antigen invades the body, there are many copies of that antigen that are all throughout the body, in body tissues and in body fluids. Some copies of the antigen are going to be present inside body cells that will provoke a cell-mediated response by cytotoxic T cells. Other copies of that antigen may be in extracellular fluid. That's going to provoke an antibody-mediated response by the B cells. So both types, cell-mediated and antibody-mediated immune responses, work together to eliminate the large number of pathogens carrying those antigenic molecules that can be in the body. Located in cell membranes of body cells are self-antigens, the major histocompatibility complex antigens. Okay, so histocompatibility, compatible with body tissues. These transmembrane proteins, remember transmembrane means that it goes all the way through the membrane. So these proteins are also called human leukocyte antigens because they were first identified on white blood cells. These are very unique to you unless you have an identical twin. Of course, identical twins have the same exact genes, so you make the same exact proteins. Thousands to several hundred thousands major histocompatibility molecules are on the surface of all of your body cells except for your red blood cells. Major histocompatibility antigens are the reason that tissues may be rejected when they're transplanted from one person to another, but their normal function is to help T cells recognize that an antigen is foreign, that it doesn't belong to you. So your immune system recognizes your MHC antigens as yours, and the immune system does not mount a response. So this recognition of self-antigens or foreign antigens is the important first step in mounting that immune response against those foreign antigens. So there are two major types of antigens, complete and incomplete. Antigens have two important characteristics, immunogenicity and reactivity. So immunogenicity is the ability to generate or provoke an immune response by stimulating the production of antibodies, specific antibodies, the proliferation of specific T cells, but most commonly both. Remember, antigen means antibody generating. So generating those antibodies in response to that antigen. Reactivity is the ability of the antigen to react specifically with the antibodies or cells that it provokes or causes to be made. So substances with both immunogenicity and reactivity are what we consider to be complete antigens. Entire pathogens, microbes such as bacteria and viruses, or just parts of those microbes can act as antigens. The chemical components of bacterial structures, the capsules, the flagellas, and the cell walls are antigenic. So are bacterial toxins. But there are also non-microbial substances that can be antigens, such as the chemical components of pollen or egg white, or incompatible blood cells, or again, transplanted tissue and organs. Just certain parts of a large antigen usually act as the trigger for the immune response, and those little small parts are called epitopes or antigenic determinants. So think of it as these are the antigens that determine what the immune response will be. So most antigens have many epitopes or many antigenic determinants, but each one of those is going to produce or induce the production of a specific antibody or activate specific T cells. So the antigenic determinants, also called epitopes, determine specifically what type of antibodies will be made or what type of T cells will proliferate to help destroy these. So antigens are very large, complex molecules. Most often they're proteins, but nucleic acids, lipoproteins, glycoproteins, and even some large polysaccharides can act as antigens. Plastics, however, have few antigenic determinants, so they can be used to create artificial heart valves or joints, and therefore not elicit an immune response. Incomplete antigens are something that we call haptens. So they're smaller substances that have reactivity, but they lack immunogenicity. So a hapten can stimulate an immune response only when it's bound to a larger carrier molecule. So an example of this is the toxins in poison ivy, which triggers an immune response only after that toxin combines with body proteins, carrier molecules. Some drugs, like penicillin, combine with proteins in the body and form immunogenic complexes, so they trigger an immune response. Hapten-stimulated immune responses are responsible for most of allergic reactions to drugs and other substances in the environment. For an immune response to occur, T cells and B cells must recognize that a foreign antigen is present. B cells can recognize and bind to antigens in lymph, interstitial fluid, and in the blood plasma. T cells only recognize fragments of antigenic proteins that are processed and presented in a certain way. In antigen processing, the antigenic proteins are broken down into smaller little peptides, and then they're associated with those major histocompatibility molecules. So think of it as you take the foreign antigens and hook them up with our own antigens so that the body can tell what's what. So once that's done, that complex of the antigen and the major histocompatibility complex is going to be inserted into the cell membranes of our bodies. This insertion of that complex is called antigen presentation. So there's a special class of cells called antigen-presenting cells that process and present antigens. These antigen-presenting cells include dendritic cells and macrophages and B cells. A lot of these are located in places where the antigens are likely to penetrate our innate defenses and get into the body, such as in the skin, the dermis, and the epidermis, in the mucous membranes of the gastrointestinal, urinary, reproductive, and respiratory system, and also in the lymph nodes. So after processing and presenting an antigen, antigen-presenting cells migrate from these tissues through lymphatic vessels to lymph nodes. So the steps of processing and presenting an antigen by an antigen-presenting cell include ingesting or bringing in those antigens by phagocytosis or endocytosis. So here we can see an antigen, a pathogen with antigens is going to be brought into the cell. Once it's brought in, it's going to be packaged in a little vesicle called a phagosome or an endosome. Within that endosome or phagosome, digestive enzymes are going to split those antigens into smaller little protein fragments. At the same time, we're going to create major histocompatibility proteins, right? So create those MHC molecules. Once the MHC molecules are made, they're also going to be packaged into vesicles. So these little green things here are the MHC proteins, and then we're going to fuse this vesicle with this vesicle. So now we've got our little antigen broken down proteins and the MHCs all together, and they're going to form a complex. Once that complex is formed, we're going to exocytose. Remember, that means to remove from the cell. The complex will be exocytosed and embedded in the cell membrane. So now we've got those antigen fragments and our major histocompatibility proteins inserted into the cell membrane. So this is presenting the antigen to T cells, and that's going to trigger an adaptive immune response. So the presentation of the antigen by those antigen-presenting cells informs or alerts the T cells that, hey, these guys are present. There's invaders, there's intruders. When you see these antigens present in the body, then you need to go into action. You need to go to combat. You need to start destroying anything that possesses these antigens. So the immune response is activated.